Thank you very much. Um, yeah, first of all, thank you for the organizers for inviting me. It's an honor and pleasure to be able to share a little bit of my research and uh, go over a little bit how we, from the paleontological side and the deep Earth history, we can use this information to potentially guide us for understanding astrobiological applications and ultimately um, to just figure out a way of just how life has evolved on our planet and ultimately, hopefully, do the same elsewhere. And so, yeah, so today I'm going to be talking about animal body size and how body size has been an important trait for animal evolution. And specifically today, I'll be focusing on how animals respond to or have responded to ancient hyperthermal events based on their body size. And so, one thing that I will just briefly go over about what I do for my research um, is so I combine several aspects of geology with paleontology to understand both the evolution of life, but also understand the physical geological context in which life has evolved. And so, because of that, I'm really interested in this coevolution of Earth and life. And so I do a combination of lots of field work to understand the rocks in which the fossils are coming from, and I also do a lot of modeling and post fieldwork analyses, using modern ecological theory to apply and understand patterns in the fossil record. And so today I'm going to talk a little bit about body size, and I will do the best that I can to explain its potential applications to astrobiological research. And I'll share a little bit of. I don't have any slides for this or for a different project I'm working on, but I'll share how body size is relevant to that. So to start, why animal body size? Why focus on animal body size? When it comes to size and when it comes to understanding our natural world, size is one of the first things that we as humans notice, right? It's very easy to compare things of completely different sizes. And as scientists and even as non-scientists, people have always been curious about large things, right? And so oftentimes people are drawn to having an interest in dinosaurs, for example, because of their large size. And on the other side of that spectrum, people have, very strong interest in understanding life at its smallest scale. And so using body size is a very easy and key way to introduce us into a lot of the organismal mechanisms that basically drive evolution and have over in time and today. But ultimately, the number one reason why we use body size, well, it's easy to measure. It's very easy to today to have quantitative data comparing modern extant species, but we also can do the same for fossil forms. So we can easily go through fossil collections, go through the literature, and we are able to measure body size and using different metrics and ultimately give us a good relational comparison with both within lineage and among lineages. And among the traits that can we can learn about by just looking at body size, those include ecology, physiology, and fecundity. There's a very strong correlation and relationship between an animal's body size and its other biological processes and its behavior. And so, for instance, one of the major groups that I work with are ectotherms, and so there's a very strong relationship between fecundity and body size among ectotherms. So the bigger an ectotherm is, the more offspring it's able to reproduce. And similarly with physiology, we know that metabolism and metabolic rates scale with body size, and that again tells us a little bit about how an organism is able to withstand certain environmental conditions. And that's going to be a little bit of what I'm going to talk about today. It's the physiological component of body size and how that responds to very ancient warming events. And so in this image here, this just documents the general history of body size evolution going back to the earliest fossil forms dating to about 3.5 billion years ago. And so we see that we have multiple orders of size change through time as we go from the very earliest single-celled bacterium to now the giant sequoias and the largest whales in our ocean. And so, and as you can see from this plot, that body size change is not linear. So it's very much contingent based on environmental conditions and also within the body plans of organisms that give them the ability to increase in size over time. And in addition to using animal body size in the fossil record, body size is also very key for today. And so another aspect of how we use body size with fossils is in terms of an organism's extinction risk. And so because, again, it's easy to measure body size throughout the fossil record, we can test the role of body size as a predictor of extinction risk in the past. And so what these two plots show here, they show the average extinction risk of organisms based on their body size over the last 65 million years. And so these are the logistic regression coefficients, and so or the log odds. And so essentially what this demonstrates is this figure, if I can put up my little laser here, this figure on the left demonstrates that over the course of the last 65 million years, on average, organisms that tend to be smaller, so those are in the negative log odds, have higher extinction risk. And when we also compare that with mass extinction events, and so you can see here, also on average, smaller body size have higher extinction risk. But what's critical about this pattern throughout the fossil record is if we compare that to today, to extinction risk of modern organisms, this pattern flips. And so, while on average in the past, small organisms have had a higher extinction risk, today it's larger organisms that have higher extinction risk. And of course, because a lot of this is driven by anthropogenic activities, essentially overharvesting, overfishing. Those are the key activities by humans that are ultimately leading or increasing the extinction risk of larger organisms. And so one of the things that I am interested in is to assess, well, if we look at various different drivers of body size evolution and body size extinction risk, can we find a pattern to dictate or to help us understand whether what we're seeing today truly is abnormal versus it being a just sort of a normal pattern and we're just missing some data? And so one of the risks of looking at the fossil record is we have an incomplete picture. So we have incomplete views of ecosystems. And so we often have to rely on what's able to preserve. And that preservation bias is going to potentially give us a skewed view of how extinction has worked in the past. And so one of the things that I'm interested in is, well, what about climate change? And so, in addition to the current anthropogenic behaviors leading to higher extinction risk among larger species, we also have a changing climate. And fortunately for us in the past, there's evidence of these short intervals of very dramatic climate change. And so, one of the things we want to see is, well, is what humans doing today comparable at all to what has happened in the past at any point? And so that brings me to the challenge of the study is, can we learn anything about both the extinction and the appearance of new species using body size for the future to determine just how much climate change is playing a role versus specific anthropogenic behaviors or direct behaviors such as overharvesting and overfishing from the fossil record to potentially inform us of what to expect in the future. And not just for the future on our planet as well, but knowing what we know about the relationship between climate and body size, can we also use that to potentially predict what kind of the size of potential life forms we might find on other planets based on that planet's temperature conditions or oxygen conditions. And so one way to, again, to be able to do that, we can focus on the fossil record. And so like I mentioned, there's been intervals of in the past of climate change, but there have also been intervals of major mass extinction events. And so, in a previous study that I published a few years ago, I assessed the role of body size in terms of extinction probability, but also the origination of new taxonomic groups. And so, in the first plot, I showed that on average, smaller organisms tend to go extinct across most of geologic time. And so in these figures here, those squares correspond to either background extinction or background origination. So these are during times where there is no extinction event or there is no major environmental perturbation. And then we have these triangles here. Triangles correspond to organisms in which their extinction risk between background extinction versus mass extinction events cannot be um, detangled. So they're basically very similar. And then we have the circles. And so the circles correspond to the extinction probability during mass extinction events, or in the case of origination, in the immediate aftermath of mass extinction events. And so in that previous figure that I showed, by Payne et al., they didn't, in that study, they didn't separate the different taxonomic groups. They basically pooled all fossil data together to look for a general body size pattern. But it turns out that that is not the most proper way to approach this because when we know from modern organisms, different animal groups have different evolutionary history that gives them a different response to different environmental perturbations. And so what I did here is I parsed out all these fossil data into their respective taxonomic groups and assessed them individually. And so what I found is that the pattern of small body size leading to higher extinction probability is generally true with exceptions. And so in this particular case, the fossil group, the trilobites, which are now long extinct, as well as cephalopods, which of course include modern existing organisms, those tend to show a different extinction probability relative to everything else that is in part consistent to what we're seeing today in the modern oceans. And so from this perspective alone, obviously these are mass extinctions in the fossil record. Humans were not around, but yet we're seeing a similar pattern. And so because of that, it's critical to figure out what we're seeing today really is human driven versus how much of that might be because in the past it's naturally occurred. With that said, what's really interesting about body size around mass extinction events is that there tends to be a much stronger response, not to extinction, but rather what new animals show up in the immediate aftermath. And so, on average, throughout the fossil record, most organisms tend to originate bigger. And so this follows an old observation known as Cope's rule that on average organisms tend to get bigger over time. And we're finding as well that even though Cope's rule referred to within lineage, we're also seeing that because of the appearance of new bigger taxa, that also is increasing the average body size. And so on average, during non recovery intervals of mass extinction events, the organisms tend to show up a little bit bigger. But what we're seeing is that in the immediate aftermath, things tend to shrink. And so that you have the appearance of smaller taxa that are smaller than the survivors. And so this indicates that the long-standing approach of using body size as an extinction probability does not detail the full picture, but instead details or points more towards origination around mass extinction events being more important. And so that's ultimately what I'm interested in is what can we predict will show up based on body size around different environmental perturbations in the fossil record. And so for this present study, instead of focusing on just mass extinction events, I focused on these intervals of major climate change in the fossil record known as hyperthermals. And so, what are hypothermals? Hyperthermals are intervals and when I say short, I mean short geologically, of usually less than 100,000 years, where the average global temperatures increase by at least 3 degrees Celsius. And so this very rapid amount of warming is thought to be too fast for some organisms to adapt to, and so could potentially lead to extinction. And so it turns out by using this parameters to define what a hyperthermal event is, we can pinpoint periods in time where we can find them. And so in addition to the rapid warming that happens during these events, there's also associated deoxygenation in the oceans. Every single one of these events coincides with some amount of widespread marine anoxia. And we're not, so this marine anoxia does not necessarily cover the entire planet and deoxygenated ocean waters, but we see large areas of it that are larger compared to non-hyperthermal intervals. In addition to that, we also see that some of these intervals coincide with ocean acidification. Some of these events have more ocean acidification than others, ultimately relating to how much carbon is sunk into the ocean, that eventually that CO2 that's drawn down will convert into carbonic acid and ultimately increase or lower the pH and increase the amount of acid in the ocean, potentially impacting marine life, those that have shelled exoskeletons. In addition to that, we also have a disruption of the carbon cycle, which in turn is partly driven by a lot of these other events that happen during a hyperthermal. And then, of course, the big thing that we're interested in about these hyperthermal events is how has it affected ecosystems, and in what way has it been deleterious to these organisms? And the reason why we're really interested in using these hyperthermal events is that we could potentially be entering our own hyperthermal event today. And so the amount of warming that the Earth has been experiencing over the last 100 years is on pace to reach, for us to reach this hyperthermal status within the next century. And so because of that, understanding hypothermals in the past can be critical to understanding our current modern ecosystem and how potentially predict how organisms will bounce back from that. And so I mentioned before, using these parameters is easy to identify several of these hypothermal events. And so far, there's been at least six distinct hypothermal events over the last 500 million years of Earth's history. These are all within the last 250 million years or so. And so of these six hypothermal events that have been observed and identified, two of them correspond with mass extinction events. And so particularly the end Permian Permian-Triassic extinction event, which is also happens to be the largest of all the mass extinction events. And immediately following that, the extinction at the end of the Triassic period. So that also coincides with a mass extinction event. These other events are also particularly well known. The early Jurassic into Torsion Pisblockian event is associated with some regional extinction. And then you have, during the Cretaceous period, two of these oceanic anoxic events, both again associated with these hypothermal warming. And then, of course, the most recent one is the thermal maximum that occurred across the Paleocene-Eocene boundary, roughly 56 million years ago. And so my goal then is to assess how animals have responded to these hypothermal events based on their body size, both from the component of extinction, but also how have new taxa bounced back and showed up after the hypothermal events have ended. And so there's been some previous work on this topic. So Carl Redden et al. A few years ago published a study in which they compared various different animal traits and to assess their extinction risk across these same hypothermal events. And so what they found is that, again, similar to what we found previously, smaller body size is associated with increased extinction risk during background intervals. And this pattern also has been shown to also occur during hypothermal events, but it's much reduced. And so, in other words, the selectivity of extinction or the probability of extinction is still the same. It's just lower, smaller magnitude of it. But again, in this particular study, they didn't parse out their fossil data based on taxonomic groups. And that's one of the things that I aim to do with this study. And so another thing to consider that I'll touch back on towards once I go over the results is this temperature size effect. And so current research around the physiological response of organisms to temperature dictates that the larger an organism is, the more susceptible it is to warming. And so in order to compensate for that amount of warming, because it again is tied to the metabolism, two things must happen. One, they either have to shrink in size, or two, they'll face the potential of going extinct. And so in these plots, they basically estimate how much body size reduction you would expect based on the amount of temperature increase over any given time. And so, and what they found using a few examples of modern organisms is that you can actually see a within lineage decrease in body size upwards of 60% from before the rise in temperature to after. And so this is a key thing to keep in mind as we go over results because it potentially explain how we're assuming extinction risk and interpreting extinction risk based on body size in the fossil record. And so with that in mind, so now we move on to the actual data part of my talk and to go over some of the methodology and the results. And so we've been using, by we, I mean folks at Stanford University, where I did my postdoc, have been compiling a body size data set for over the last 15 years now. And this body size data set consists primarily of body size of all marine animal genera that we know from the fossil record. And so there's roughly about just under 60,000 identified genera of fossil marine animals. And so our goal is to eventually be able to assign a body size to every single one of these marine genera. And so right now we're at about 34, 35,000, and so a little bit over halfway. And so work continues. And so the way we've actually collected these data has been pretty great and a great experience, not just for me, but they also for the actual data collectors. And so one of my jobs as a postdoc while I was at Stanford, I ran this summer internship program for local high school students in which they would come in for 8 weeks at a time. And basically, they would get hands-on experience with collecting data. And so we used several compendiums, particularly the treatise on invertebrate paleontology, and we used calipers to measure the images of fossils in these compendiums. And so based on these measurements, we then tabulated all of that into a large data set. We calculated the volume of organisms based on these linear measurements, and we've compiled our data set. And so this program went through a hiatus for a couple of years, but it returned last summer and will return again this coming summer. And so at first glance, you might think, oh, we're bringing in these high school students and they're just collecting data. It seems kind of exploitative. Well, it is in a way, but the students do actually get a lot out of this. So, in addition for of them collecting data for us, we also teach them quite a few things and give them their own research experience. And so, one of the things that when I was running the program, I would give them a couple weeks of lectures and activities in which they learn a little bit about natural history, learn a little bit about the biology of body size. And then I gave them a very sort of short introduction to using R and coding, and also getting them introduced to using quantitative methods. So very simple statistical methods and tests that they can learn and apply to different things. And so once they have that very general information down, we then give them the option to work together in groups to work on a research project. And these research projects are actually using real data And so they are proposing their own questions that have, not been published before, and to figure out what are some patterns they can tease out of the data that they themselves have been collecting. And so while, for us, it's a huge benefit that they've been collecting these data for us, they've also got quite a bit out of it. And in many cases, we had them, um, as they're working on their projects, write up an abstract for AGU. And so before, for those of you who attend the American Geophysical Union conference, it used to be regularly in San Francisco, which is only about an hour's drive away from Stanford University. And so we would basically take them in a van and have them present their research at the conference. And so they got quite a bit out of it. And on the other side, we ended up with lots of body size data. And so here's very quickly just how that body size data is collected and how we use it. And so, as I mentioned, we use calipers to measure the images of fossils from the compendium, or if there aren't any images, if there's any actual dimension or measurements listed, they'll take those. But ultimately, we're looking at taking the three major axes of any fossil organism. So it's height, length, and width. And so wherever possible, we use those three axes. But in most cases, because we're dealing with images, we can only ever get height and length. And so using a logistic regression, we can then predict the width based on other data we've already collected. And so using these three axes, we then calculate volume of these organisms. And so this is a very rough way of doing this, but for organisms, for most fossil organisms that we dealt with, we basically assumed that they were a sphere-like shape. And so we use the formula for the volume of a sphere. And for fossil forms that tend to be coiled, things like snails or ammonites, we'll use a cone approach. And while this may not be the most precise way of measuring volume for these organisms, because we ultimately log all data, the difference, the margin of error is pretty small. And ultimately, what we're more interested in is whether these data are enough to tell apart among lineages versus within lineages. And it turns out, after doing quite a bit of sensitivity analyses, this approach has been fairly robust. All right. So this is what the data look like. And so the first publication of these data happened 10 years ago. And what this plot shows here is the average body size of major marine animal groups based on their phylum. And what this shows is over the last 550 million years, on average, body size has increased over time. And what each of these little individual line or segments correspond to, it is the duration or the stratigraphic range of individual genera, and they're color coded based on their phylum. And so early on in Earth's history, we were primarily dealing with smaller invertebrates. And so you can see today that the largest organisms in the ocean tend to be chordates, so vertebrates, so whales and other marine mammals. Oop, let me go back. And so the total data set that we're looking at are 6,000 genera of solitary organisms. And these includes things like bivalves, clams, fish, cephalopods, echinoids or sea urchins and sand dollars, snails, and brachiopods. And if you're not familiar with what a brachiopods is, it's an organism that superficially looks very similar to a clam, but is very, very distantly related to them. And they have, and for that reason, they have different metabolic rates. They're still around today. They're just not as abundant as they were in the fossil record. And so, as I mentioned, we use volume, and so we log that volume. And in order to have a robust enough sample size, I focused on groups of organisms with at least a minimum of 200 genera with body size. And so each of these six groups have at least a minimum of 200 genera. And we focused on the interval from the Permian to the Pleistocene. So the Permian leads up to the first hyperthermal event at the end of the Permian. So we wanted to include that background information all the way to 1 million years ago. And to do all of this work, we took fossil occurrence data from the Paleobiology Database. The Paleobiology Database is a freely available online resource where published material of published fossil material is uploaded, and it's based on geographic occurrence. And so we can able to plot where they were found using both modern GPS coordinates, but also more older historical location data. And we're able to put enough data together to have a somewhat of a big picture of what's happening around the world. Unfortunately, because of several biases, and particularly around the global north versus the global south, most of the fossil record from the Paleobiology database is overrepresented by the global north. And so as you look at these data, there's something to keep in mind. It's that it's mostly giving you a picture of the northern hemisphere, not so much about the south. And so it's one of the things that collectively within paleontology and paleobiology we're trying to address. But for the time being, unfortunately, this is what we have to work with. And so we'll start here and hopefully we can expand on that going forward. And so the method that I used to calculate extinction and origination probability is known as capture-mark-recapture. And so capture-mark-recapture comes from modern ecologists, in which they basically do a census count over a certain habitat, and they come back multiple times to determine what is the history of that population in that area. And so effectively, what happens is you go to a certain area multiple times. And so when you go the first time, whatever you find, you capture it, you mark it, and then you release it. And then when you come back again, you try to look for it to see if you can find it. And so, and then there can be times when you go back and I don't know why this is going on its own. I apologize. And Stop. There we go. So as for every attempt in which we look for that original specimen, we mark it, and so we give it a one. And in cases where we go in and we don't find it, we give it a zero. And so there's two probabilities here. So one of them is that the organism that we capture the first two attempts, and we didn't see it in the third attempt, perhaps it died, or it just happened that we didn't see it that time. We just didn't sample it. And so this zero can correspond to either it died or we just didn't sample it. But in this case, since we saw it again, then we can determine, yes, this zero is because we just didn't sample it. And so, and we take this history, and it's basically a history just binary values, whether they're present or absent. And so between each interval, we calculate the probability of will it survive into the next sampling attempt, and what is the probability that if it did survive, that we're actually going to encounter it. And so we can do this for both extinction and origination. And so we can actually figure out when new things show up and when they were not there before. And so using this approach, we then have this long, for each individual genus, we have this history of appearance and of occurrences. We have the probabilities of whether or not they died or showed up in between sampling occurrences, and whether or not we actually were there to sample it. And so in this particular case, the sampling attempts correspond to geologic time bins. And so, we're using the highest resolution time bin available for us, which is, I am so sorry, which is the stage. And so basically each of these numbers correspond to a single geologic stage. And since we are covering the Permian through the Pleistocene, that equates to about 50 stages. So we have 50 time bins of data. And we have a little over 6,000 genera. And so essentially we have a list of 6,000 binary strings. And based on those strings, we can calculate probability between each interval and also consider body size. And so we put all those probabilities as the dependent variable into logistic regression models in which we look at time and size. And so we have to compare normal background intervals to those during these hyperthermal events. And so background intervals is just this additive model, time plus size, whereas in during intervals of hyperthermals or the recovery intervals immediately afterwards for the origination, we use a multiplicative model. And so all told, there was 96 models that were fit over on 6 taxonomic classes. 16 for each class, both 8 over extinction and 8 over origination. And so these models all correspond to, in addition to body size, we also use other covariables such as geographic range and a few other things. But it turns out that in each of these cases, body size tends to be the biggest or the strongest indicator of extinction and origination. And so we're sticking with body size. And so here are those actual results. And so looking at extinction first, and so the orange squares here correspond to the background extinction, and the circles correspond to extinction during hyperthermal events themselves. And so the big takeaway here is that once more, we see that on average during background intervals, smaller things tend to go extinct. And during these hyperthermal events, most organisms don't differ very much from their background risk. And so we see that here that in most cases we have a strong overlap. The only two exceptions to that are fish and the brachiopods. And so brachiopods, those that are tend to be larger, have much higher extinction risk. And whereas for fish, the background probability of extinction is amplified during hypothermal events. But all in all, we don't, just generally speaking, we're not seeing a major effect of extinction around these hypothermal events using body size. But if you look at origination, however, we see a completely different result. And so once more, as I mentioned from the previous study early in this talk, on average, larger organisms tend to show up during background intervals. So you see these orange squares here. But during these hypothermal events and the recovery of them, they actually show up smaller. And so on average, so we have these three groups that show up smaller. This one shows up smaller. This one is pretty much doesn't change a whole lot. It's kind of random with regards to body size. But when it comes to bony fish, this is again the one outlier where clearly large fish tend to thrive in these environments. But overall, this is the pattern we're seeing. So smaller organisms tend to appear newly in the fossil record immediately following these hyperthermal events. And so now the question is, well, why is that? And so based on the temperature rule that was put together by Curtis Deutsch, we should expect to be higher extinction of larger body size organisms. We expect to see a lot of this across all different taxonomic groups, but we're just not seeing that. And so, and this, of course, does actually make sense because uh, it makes sense because after these events, organisms tend to have to shrink, and the new genera that evolve from those existing survivors will tend to be smaller on average. And so, and just to further check on these results, so we looked at the model weights of for extinction origination. So going back to just really quick, this model here. And so we look at so this model here corresponds to you both have background and a hyperthermal extinction, which means that you have two separate extinction or intervals of extinction risk, and so or origination risk. And so this is what these plots show is that for when it comes to extinction, um, these having hyperthermal in the model doesn't really give it much weight, with the exception of bony fish. Some weight for cephalopods and rhynchonellates, but for gastropods, echinoids, and bivalves, hyperthermals make no difference in terms of their extinction probability. Whereas if we look at origination, so the appearance of new organisms following a hyperthermal event, it clearly has a very strong support. And so for all of these groups, even ones that experience no big change based on extinction, when it comes to origination, you see a very strong response in how they respond to extinction. So it clearly affects hypothermal events, clearly affects how new organisms show up, but it has very limited effects on extinction, all based on body size. And so to get to the potential explanation of why, and so I mentioned before that from Curtis Deutsch's work is that one way to compensate for the increasing temperature is to decrease in size. And so one possibility we're seeing here, and it's looking to be the most likely explanation, is these organisms are not going extinct based on body size, as you see here. They're basically in some of these cases, it's completely random in regards to body size because they're adapting to that change in climate. And so, even though these are very rapid events, it turns out that we actually have some data that support that certain organisms can, in fact, change in body size within human lifetimes. And there's a lot of examples of this from archaeological data, where we look at shell middens or the discard piles of indigenous groups and once they feed off shellfish, that if we compare the size of those shells with the ones around today, the ones early on were bigger than they are today. So they, within a span of a few thousand years, organisms were able to shrink to adapt and avoid being basically harvested by early humans. And so, based on that, it's entirely possible then that these organisms were adapting by shrinking, and so to basically just write out the hyperthermal events, and because they decrease to a small size, they likely stayed at that small size. And so when they speciated into a new species, into a new, and also into a new genus, they kept that small size, which would explain why in the aftermath you'd have the appearance of smaller taxa relative to those that survived. And so, with all that said, it turns out that extinction is less selective with respect to body size across hypothermal events, but origination is by far really strongly selective during the recovery interval. And so these results suggest that warming and deoxygenation alone don't have a major effect on extinction with respect to body size, which means that for the modern climate, it's likely not playing a big role, and it's really just mostly from humans. Um, Climate-associated perturbations do change extinction and origination compared to background intervals. So climate change does have an impact on ecosystems regarding body size. It just is not always the way that you would expect. And then lastly, based on these results, any sort of climate-associated evolutionary dynamics in the modern and future ocean and potentially in other planets, we can find that they can differ from regular or normal, what's considered normal geologic background patterns in potential predictable ways. And so knowing how modern organisms respond to climate conditions and warming and other associated climate stressors, we can then potentially predict what kind of animal body size we would expect to find, or organism or life form size we would expect to find on other planets based on that planet's climatic conditions. So with that, just very quickly, acknowledgements, lots of folks to thank, and I'll just leave it there. So thank you so much.